On behalf of the National Stem Cell Foundation of Australia, welcome to Future Medicine, Stem Cells and Women's Health. My name is Tanya. I'm Director of Engagement at Science in Public, where I've looked after communication for the Stem Cell Foundation for over 10 years, and I'm delighted to be your host this evening. I'd like to begin by acknowledging the traditional owners of the lands from which we're joining this event. I'm coming to you from the lands of the Wurundjeri people of the Kulin Nation. I pay my respects to their elders past and present and recognise any people from other First Nations who might be joining us. Before we kick off, I'd like to note that we've called this event Stem Cells and Women's Health, but we're using the terms women and girls for convenience, but we do wish to include people assigned female at birth and or those who have a uterus, and also acknowledge that men can get breast cancer and endometriosis too. Having said that, it is tough being a woman, especially when it comes to health. Endometriosis is a painful chronic condition. In Australia, it affects at least 1 in 9 girls and women. It is often dismissed as just painful periods, and on average, it takes 6.5 years to be diagnosed. Pelvic organ prolapse is a debilitating condition that affects many women soon after or many years after vaginal birth. There is currently no reliable cure. And breast cancer is the most commonly diagnosed cancer in Australia. In 2022 alone, 3,178 women and 36 men died from the disease. But the good news is that women's health is getting more attention and better treatment options thanks to the work of scientists and clinicians in Australia and around the world. The National Stem Cell Foundation is a health charity with 2 key missions. One mission is to support the stem cell scientists doing evidence-based research. Our other mission, and the reason we're here tonight, is to provide the broader community with information about this emerging field of regenerative medicine so that they can make informed choices about their own healthcare. Tonight, we're going to hear from 2 of our directors who are stem cell scientists and also a leading bioengineer. The work of our panellists is paving the way for new therapies and a better understanding of health conditions that disproportionately affect women. Our panellists are Professor Caroline Gargett, who is studying endometrial stem cells and their role in endometriosis, Dr. Shayanti Mukherjee, a stem cell scientist and bioengineer who's developing treatments for pelvic floor injuries, Professor Geoff Lindeman, a clinician scientist treating cancer patients and researching breast cancer stem cells and their role in cancer. We will hear a short presentation from each of them shortly, and I'll ask them a couple of questions. Later, we'll bring all of the panellists together and you'll have your chance to ask your own questions. Please write them in the Zoom chat box and we will get to as many as we can. And behind the Science in Public participant logo is my colleague Kieran. He says hello. He's providing tech support. You can send technical issues to him directly via the chat function. Also note that this evening's event is being recorded so that we can share it online at a later date. It now gives me great pleasure to introduce our first speaker. Caroline Gargett is world-renowned for discovering stem cells in the human endometrium, the highly regenerative lining of the uterus, thereby establishing a new field of research in reproductive biology. She heads the Women's Health theme and the Endometrial Stem Cell Biology Group in the Ritchie Centre at Hudson Institute for Medical Research. She's also a director of the foundation. Over to you, Caroline.
Tanya. Yes, thank you, Tanya, for that kind introduction, and welcome to you all, and thank you for joining us tonight. Before I start about talking about what about endometrial stem cells and endometriosis, I would like to just first acknowledge members of my team, both past and present, and also our major funding bodies. Oh, I've got a technical issue. Why is it not going? Oh, here we go. Now, before I talk about endometrial stem cells, I want to firstly just give you a little bit of a brief introduction about what adult stem cells are, because that's what we're really talking about tonight. Adult stem cells are present in most tissues and organs of the body. They're very rare cells, but they're quite unique and different from all the other functional cells of any tissue or organ. They have several properties. These unique properties are that they can undergo what we call self-renewal and produce identical copies of themselves. But at the same time, they can differentiate. And just a minute, I'm going to get my mouse. They can differentiate and produce cells that proliferate a lot and generate the functional cells of the tissue. So a single adult stem cell division can produce this, what we would call a clone of tissue or a set of tissue in a particular organ. So to identify the activity of endometrial stem cells in the lining of the uterus, we looked at these adult stem cell functions. And so we found, we took endometrial cells from endometrial tissue and we looked at their ability to make clones. And you can see on these plates here, epithelial and mesenchymal clones started by single cells. And those clones, which is like producing this cluster of cells over here, they can also self-renew, and the epithelial ones will produce large gland-like structures when they are made to differentiate in vitro. The mesenchymal cells, on the other hand, they can also self-renew and they can differentiate into mesodermal lineages. And from this work, we concluded that human endometrium contained rare populations of epithelial progenitor type stem cells and mesenchymal stem cells. But the problem was we had no idea where they were. So we set about identifying their niches and looking for markers of those cells that would tell us where their location was. And this is just to remind you that the endometrium is the inner lining of the uterus. And this diagram here is very much a schematic of a unit of tissue. It's a gland that extends from the lumen right down to the muscle part. And there's thousands and thousands of glands in the endometrium. And basically this tissue, this yellow part, is shed every menstruation, and it leaves behind a raw surface, a small amount of tissue called the basal layer that is resurfaced by these green cells. And I'll talk more about them in a minute. So the markers that we found that identified these individual cells with the properties I've just talked to you about before, for the mesenchymal stem cell is SUSD2. And we found that these cells were around blood vessels. They're in the vessel wall, in that layer that's shed during menstruation, and also in that layer that stays behind. We also found, in another project, we found markers for what we call the epithelial progenitor stem cell, which is down here. It's called N-cadherin, and it's right in the very bases of these glands. And in the basal layer, these glands are sort of quite horizontal and interconnected. So they're right down near the uterine muscle. We also found another type of progenitor cell called marked by the marker SSCA1. And these are the cells that are at the right spot when the endometrium's shedding off, it rapidly resurfaces and repairs that endometrium. So since we've had those markers, we could now look at their role in endometriosis. And Tanya has already introduced a little bit about endometriosis, but basically it's the growth of endometrial-like tissue outside the uterus. It's very common, as Tanya mentioned, 1 to 7 to 1 in 9 reproductive age women, 1 in 2 infertile women, and 1 in 2, or even more than 1 in 2, in women and teenage girls who have severe pelvic pain. But it's outrageous that it takes 7 to 10 years to get diagnosed. And this is what we want to change. And what we're trying to do is look at the role of adult stem cells, endometrial adult stem cells in endometriosis, really trying to understand the cause. And one of the things that we've done to do this is we've looked in menstrual fluid. And because we've got those markers, we could actually measure the concentration of the SUSD2 mesenchymal stem cells and the N-cadherin epithelial progenitor cells in women with and without endometriosis. And we found there was not different at all. They were quite similar, and there were quite a lot of cells there. But not all menstrual fluid comes out through the vagina. Some goes back through the fallopian tubes and gets into the pelvic cavity. Now, this happens in almost all women. So we were thinking that in women who get endometriosis, maybe it's the stem progenitor cells that get access to that pelvic cavity. So we did this hard experiment where we took women who are having laparoscopic surgery on day 2 of their menstrual period, and we collected the peritoneal fluid. And again, we could measure that the SUST2 EMSCs and the encadherin cells, and we found that they were indeed increased in women with endometriosis. So those little fragments of cells containing those stem cells can land and adhere to the peritoneal lining. And just like I showed you right at the beginning, they could actually proliferate and form like a clone of cells, actually like a little unit of tissue, which is like the endometriotic lesion. We also looked in peripheral blood, but we really hardly found virtually no endometrial stem cells circulating in peripheral blood. But I also want to just mention about menstrual fluid, because it's a non-invasive source of endometrium or endometrial tissue. And here's just a smear of menstrual fluid, and you can see gland fragments floating around in a sea of red blood cells. We can collect it with a menstrual cup. We can make organoids from these gland fragments, and indeed we have. And these organoids are like 3D cultures of the epithelium, and they really are like mini glands, okay, that I was showing you before in that schematic. They're very similar to the endometriums, the ones that we get from surgical laparoscopies. And we also find that the SSCA1 cells are present in those organoids. But we've also looked at the composition of the menstrual fluid, and we found that it's relatively constant in any individual woman on consecutive menstrual cycles. So we think it's really quite an important biofluid, and that's what we're trying to develop for diagnostic purposes. We could look at the concentrations of the adult stem cells that we've talked about, and endometrial proteins and exosomes as biomarkers for endometriosis. But we can also use the organoids that we can get from that menstrual fluid, and we can develop patient-specific organoids, and we could test them with drugs or hormones before for women before they actually try them for treatment. It's like try before you buy in personalised medicine, because not everything works for every person. We're also using those organoids to understand the infertility that's associated with endometriosis. And in a non-stem cell project, we have another area that we're looking at the effect of a new interferon for treating endometriosis. And just to finish my talk on the research part and looking at the cause of endometriosis, what we're currently doing now is investigating the role of the endometriosis risk gene. So this is really looking at the genetic effects in endometrial adult stem cell function. And with that, I would like to thank you for listening, and thank you very much. And I can share.
Thanks so much, Caroline, for that. I'll ask a couple of questions now, and it will be your turn, audience, to ask questions when Caroline joins us later for the panel discussion, but you can put your questions into the chat box in the meantime. What I wanted to know was that ability to produce, the organoids in the lab, so to recreate endometrial tissue in the petri dish, so to speak, is that something that's going to be useful for, say, screening different treatments to see how, other treatments that are used for different diseases, drugs, for example, is that going to be a useful way to test them? Because those drugs have already gone through safety tests.
Absolutely, and that's the type of thing that we're wanting to try and do. The organoids really has changed endometrial biology. It's fairly recent for that technology to have arisen, but it's really incredibly valuable, and being able to use either endometrial tissue itself from surgical samples or menstrual fluid makes it really easy, especially the menstrual fluid, to generate those organoids, and that you can culture them and you can passage them, and you can, once you've cultured them initially, you can actually set them up from single cells as well. And we think that probably they've got some stem cell function in those cells that initiate an organoid on its own.
And how confident are you that those living cells from the menstrual fluid, from the tissues that you've cultured and from the organoids, how confident are you that they will behave the same way or a similar way in the patient to the way they behave when you're using them in the lab?
Well, that's a $64 question, and it's quite important. And that's where a lot of research is going on at the moment. So, for example, with our project where we're looking at the effect of the hormones and infertility in endometriosis women, it's really looking at the hormone effects. We can see that the hormones are having similar effects on the organoids as they do in the women, for example. So we can recapitulate some of the pathologies, I suppose, that occur. So that's how we can tell, is if we can recapitulate what's been found already, or what people experience with endometriosis, then we know that they are a good model. But in other systems, they have been a good model too, in other tissues.
Yes. And I guess it is relatively early days with endometrial stem cells compared to, say, kidney organoids and cancer being recreated in the lab. Well, that's all we have time for at this stage, but we will have Caroline back with the other speakers for the open discussion later. So do send through your questions. Our next speaker is Shayanti Mukherjee. Shayanti is head of Hudson Institute's Translational Tissue Engineering Lab. She specialises in innovative cell-based therapies for pelvic floor disorders and birth trauma injuries. Over to you, Shayanti.
Hi, everyone. Can you hear me well? I hope so. So I'm a bioengineer, and I had no idea about women's health or stem cells. There was a time where I came across stem cells as a very fascinating new area of research. And then independently after my PhD, I came across women's health, and I was quite sad to know about this life after having child for a woman is not as glorious as it looks in a lot of the pictures that I see. I thought life would be great, but sometimes it's not. Sorry, can't navigate. Okay. So today I'll be talking to you about pelvic organ prolapse and what that is. So it's the downward descent of pelvic organs such as the uterus, bladder, or rectum into the vaginal wall, or sometimes outside the body. And as you can imagine, this could be quite a difficult life situation for women. And sadly, this is a very common situation. And about 1 in 5 women would need reconstructive surgery. And the failure rate of these surgeries can be quite high. So as you can see in this picture here is like, imagine that, that our pelvic floor and the muscles there are like a fruit bowl, and it's kind of holding all the organs together. And when women undergo childbirth, there's excessive stretching of the tissues, and sometimes they lose their strength. And so what happens is the mechanical stability is sometimes compromised, and the organs kind of fall out of their original position. This leads to quite poor quality of life for women. So they can have urinary, fecal, and sexual dysfunction. And these are it's quite a taboo subject. And so women tend to live many, many years in silence. They suffer in silence, and it significantly impacts their life. And what is really difficult is there's no optimal therapy at the moment or cure that can be a one-stop solution. So there's a lot of inadequacies in the treatment that we have. So how does pelvic organ prolapse affect women? So this figure was actually generated by Lynn Ledger, who is a great advocate in this field, and she is a fellow Australian, and she worked with us to make a diagram out of what pelvic organ prolapse and avulsion, which is the tearing of the muscles when women give birth, how does it impact? And as you can see, the impact is wide, and it affects every part of a woman's life. And so for women who actually have a high degree tear and prolapse, going to work and living life is quite difficult. Why does this happen? So let's have a look at why this happens. So we all are quite aware of vaginal childbirth and that there is excessive stretching. So imagine, so this was a computational model that was developed by one of the greatest scientists in our field. This blue ball here actually represents a baby. And these pink lines here represent the pelvic floor muscles that hold the organ. And this little red ring here represents the vaginal opening through which the baby comes. So you can see that for this baby to come out of the vagina, the muscles need to stretch quite intensely for it to come out of the vaginal opening. And so when it is at full dilation, some of these pelvic floor muscles actually undergo a stretch of over 3 times its capacity. And based on this computational model, they actually showed that it was like the model said it's probably not going to happen. It's not physiologically possible, but we know it is possible. But what this tells us is if our leg muscle undergoes that kind of stretch, we're probably going to land up in hospital with severe injury. But childbirth, pelvic floor muscles are quite unique in their adaptation. So what this says is there's a massive risk of things going wrong, overstretching, which compromises the stability of the pelvic floor as the childbirth happens. And over the lifetime of women, factors such as multiple births or obesity, aging, they make things worse. And so there's a high chance of women getting prolapse. So what happens when she gets prolapse? So in the past, so there are a few treatments. So you could take sutures and reinforce the tissue, which is called the native tissue repair surgery. And a lot of you would have heard about the mesh debacle, where synthetic non-degradable mesh was taken to augment these surgeries to provide that additional strength because there were so many women in need. And these were introduced without proper trials, proper preclinical or clinical trials. And after several years, what happened was there were more rates of complications, and ultimately the products were withdrawn and banned in several countries, including Australia. So why did this happen? This happened because these materials were not tissue friendly. So for any material that goes inside our body, we want these materials to be working with the body and not against it. So we want to design better materials because there is a huge unmet clinical need right now. There's a growth in aging population, and we are going to have more women needing these surgeries. We have more women wanting a normal life. And so we need to develop and work with the body, understand why the body is not accepting such measures, and what can we do differently to make the treatment better. So having a look at this tissue. Okay, so I'm just going to take you through a microscope. So here what you see is the blue color is actually a histological section of human vaginal tissues. And if you zoom into this little square here, you see this structure at the nanoscale. So this is actually the collagen in the tissue that makes up that provides the strength to the tissue, and it looks a little bit like the corn on the cob. But what this tells us is the tissues are made up of little molecules of nanostructures. So in order to provide better treatment, we need to mimic the tissues better with better materials, and two, we need to provide them with cells that complement and repair and regenerate rather than impact the body negatively. So what are we doing about it? So Caroline gave us a beautiful outline of what endometrial stem cells are and how they are being used in the diagnosis of endometriosis and their treatment. We are using endometrial MSCs for a different purpose. So Caroline has discovered endometrial MSCs, and she's shown that these can be taken out from the uterus. These are the cells that repair the uterus beautifully to perfection each month. So can we take these cells out of the body and use them for the treatment of pelvic floor? Now, we've developed this sort of program where we are trying to take these cells out of the body and use external, more tissue-friendly material to regenerate the pelvic floor. So our first program is 3D printing and nanotechnology. So here we are using advanced technology. So there's so many new things happening, and medical treatment has been revolutionized using technology. Then why not women's health? So we're taking advances in 3D printing and nanotechnology using degradable polymers, providing that ultrafine structure that body has to mimic the body structures more efficiently and combine them with women's own stem cell. So imagine there is a surgical suite where there is a 3D printer, and we have a scan from a woman that helps us provide the design of what kind of mesh it should be. The machine can print that mesh of ultrafine structure. It can also print women's own stem cells within the same equipment on the benchtop, and there it's ready for surgical application, personalized completely for a woman's her own needs. And so this strategy we're using as a treatment that could be for suturing and treating pelvic organ prolapse in its chronic stage. The second therapy is these amazing stem cells, endometrial stem cells, which can regenerate uterus to perfection. Why not use them for preventing pelvic organ prolapse? What if we could take them before women were planning and there was a risk of pelvic organ prolapse? So we could actually combine them with hydrogel. And if she has an injury after birth, we could actually inject them soon after birth. Why wait until she develops prolapse later in life? Why not do something soon enough? So a big part of our program is looking at various aspects of different disciplines, which is looking at the immune system, how the body reacts to such construct, what material should be used, how should the design be, is the tissue regenerating properly, and what about the stem cells? How can we make them more usable, more efficient in doing what they are capable of? So a big part of our program looks at combining these different disciplines and bring together technology to have a one-stop solution for pelvic floor disorders. I imagine a future where women give birth, and if they have an injury, they could actually go to the hospital and get a treatment and get on with life. Okay, so this is the future that I imagine. Thank you so much.
Thanks so much, Shayanti. That's both terrifying and exciting. I found that image of the baby that looked to me like a bowling ball, kind of relatable given how large my second born was. But look, I think it's exciting to see, but it is the future. How far off do you think both the polymer, the degradable polymer mesh, bioengineered tissues and injectable cells, how far off do you think we are from seeing something like that in the clinic?
Look, that's a very good question. I sometimes ask that myself. If I had everything that I needed for this project, I would say 5 to 8 years, because we want to ensure that we do the proper trials. Last time they were, used without proper trials. It kind of has set the field back significantly, and the sufferers are women who have the disorder. So if I had all the resources that I need and all the necessary experiments, I would say 5 to 8 years, we should be ready to have a proper clinical trial protocol. Yeah.
That's excellent. And in the meantime, we should do our Kegel exercises. Is that right?
So I think the treatment for pelvic organ prolapse, so exercises, pelvic floor training is recommended as a conservative treatment until they are actually eligible for surgery. So while it can delay the progress, unfortunately, they cannot avoid or solve pelvic organ prolapse. So yes, it is definitely important, but what we need is a treatment solution for women who are not fortunate enough to live a good life with pelvic floor exercises there.
Okay, well, that's all we have time for at the moment, Shayanti, and I can see
you.
that, Ulla, Sam, Pesman and a few others have started sending through questions. So you can have a look at those, you and Caroline,
Sure.
in the
Thank
chat
you.
and have a think about those, and we'll get to those when we have our open chat later on this evening. So thank you, Shayanti. And that now brings us to our final speaker, Professor Geoff Lindeman. Geoff is a clinician scientist investigating normal mammary gland development and events that go awry that lead to breast cancer. Geoff is joint head of the Cancer Biology and Stem Cells Division at WEHI and a medical oncologist treating breast cancer at the Peter MacCallum Cancer Centre and Royal Melbourne Hospital. He's also one of the foundation's directors. Over to you, Geoff.
Thanks so much, Tanya. I'll just, I hope I'll share my screen. Can people see that? Can you see that?
Yes, that looks fantastic.
Oh, okay. Look, thanks so much for the opportunity to, I guess, look at another area of important health for female. And as you've heard, I'm at the Cancer Biology and Stem Cells Division at WEHI, and I want to begin by acknowledging all of the people that actually do the work, and in particular Jane Visveda, who's I've worked with for many years now, and who's very much a mastermind of a lot of the work that I'm going to discuss. But I want to, I guess, just take a step back and reflect. We're talking about stem cells in adult organs, and really there are very many shared themes across organs that we should consider. And in one sense, the blood development system has been provided a paradigm for a lot of other adult organs like endometrium, like breast and other tissues. And as you've heard, we have a succession of stem cells that can divide into daughter cells, become more restricted and differentiated to really then end up as mature workhorses that provide the tissue for that organ. And this is not simply conceptual. We know that, for example, for the blood cancer area, Don Metcalf, who was at WEHI for over 50 years, worked with many colleagues on colony stimulating factors, and these really have come to the fore in terms of our understanding and treatment of cancers. And in fact, more than 20 million people have now benefited from having their blood counts boosted during cancer therapy, including breast cancer therapy, and also for bone marrow transplantation. So turning to breast cancer, it's obviously not a trivial problem, as Tanya introduced. We have more than 600,000 women each year dying around the globe. In Australia, at least 9 women a day are now dying from breast cancer. And although the news is really good in terms of improved survival for any woman who's been diagnosed with breast cancer, those incidence rates keep rising. Of course, it's going to be multifactorial in terms of the conditions that and background issues that give rise to breast cancer. But it's fair to say that our understanding is sort of underpinned by a combination of the roll of the dice, the fact that breast cancer happens much more frequently to women at older age of onset, and family history with genes is very important. And of course, reproductive factors such as female hormones are very important. And I've just put this up so that we can perhaps have discussions at the end. So what Jane and I and people in our lab have tried to understand for many years is what are the cell types that actually give rise to breast cancer, because it's not simply one condition. The pathologists describe at least 20 types of breast cancer, and we know when we look at a genetic level that they can be stratified into at least 3 or 4 major subtypes. We want to understand what role female hormones play, and most importantly, through understanding normal development and stem cell biology, how those findings can be applied to treatment or prevention of cancer. And conceptually, breast cancer by and large part arises in the ductal tissue. These are the epithelial cells that form a bilayer of cells comprising the inner luminal cells and the outer myepithelial cells, which have muscle-like function and protective function that can squeeze milk through the breast ducts. So it's these ductal cells which undergo massive changes during a woman's life, and in fact with each menstrual cycle, that are the culprit cells that can give rise to breast cancer, which is thought of as being a succession of events where there might be early changes through genetic lesions that lead to a precancerous change that can then lead to an invasive breast cancer. So we want to understand these events and how all of these cells give rise to the tissue in the breast, and whether there are certain cells which are more prone to breast cancer, and therefore how we can intervene. And so the stem cell, as I've mentioned, is going to be the mother of all of this mammary epithelium. It will give rise to a succession of cells that form that bilayer. And it's important to really understand whether the genetic events happen in a stem cell or whether they might happen in a daughter cell that sustains that mishap. And that's one of the areas that we've got a strong interest in establishing. And one of the things that our group has contributed to is the identification of a hierarchy of cells in the breast tissue that starts with a stem cell, which is capable of giving rise to all of these cells through various assays that can differentiate into these progeny. And we've done this work in both mouse, but also in human breast tissue from women who donate tissue through reduction mammoplasties. And through that, we've identified a stem cell containing basal population, the daughter cell or progenitor population, and a variety of mature cells. Interestingly, when we looked at the way that these cells express or display their genes, they very markedly reflect the different types of breast cancer that we see in the clinic. We know that the progenitor cells, the luminal progenitor cells, look very much like what's called basal-like or triple-negative breast cancer, that is a very clinically aggressive type of breast cancer that can rise in patients, whereas those hormone receptor positive breast cancers look much more like the ductal cells. Now, is this actually relevant for women who are at high risk? And that's one of the first questions we started years ago once we were able to catalogue and prospectively isolate these cells. And one of the conditions that many people will be aware of and made famous by Angela Jolie is people who carry a faulty BRCA1 gene, who have a particularly high lifetime risk of developing breast cancer and also ovarian cancer. And the types of cancers that they develop are these very explosive triple negative type breast cancers, which are negative for many of the markers that we use as targets in the clinic. So really all that's left for these patients is chemotherapy at the moment, plus some other emerging therapies. So once we had been able to identify various subtypes of cells, we then looked at BRCA1 tissue from patients who were undergoing preventive mastectomies, and Elgene Lim and then Emma Nolan in the lab were able to discover that it's these progenitor cells which are most like basal-like breast cancer and also misbehaved in some of our clinical assays and were almost certainly the cell culprit cell that gave rise to breast cancer. This at the time was quite a surprise because people had expected the stem cell, rather than the daughter cell, to be an important player. But Emma Nolan, when she was in our lab, was able to further fractionate these stem cells and identify a marker known as rank, which is very important for bone biology and the immune system, but it's also present on these progenitor cells, and really was a flag that identified these cells as being the most at risk that are cancer prone for BRCA1 mutation carriers. Rank is very important in the clinic. It's used as a target for people with osteoporosis because there's a drug that can actually switch off this pathway called denosumab. So we have looked at whether or not we could switch off rank signaling in these breast cancer prone cells, and that's indeed what we have found. What we think happens is that this rank positive cell, which has problems with repairing DNA and is highly proliferative, can very rapidly undergo cancerous change, giving rise to breast cancer. And some of the preclinical work and work with human breast tissue that we've done suggests that switching off this pathway might be a way to actually curb these cells even before cancers arise. And as I've mentioned, there's a drug called denosumab, which is already used in the clinic. So our hope is that that could be repurposed as a breast cancer prevention drug. And that has led to an international prevention study called the BRCA-P study, which is now being undertaken across 7 countries, including Australia, where we are randomizing patients for therapy with denosumab, which we understand the safety profile for, and placebo, to see if we can reduce breast cancer events and also look at other secondary endpoints. And people who are interested in exploring this or further can look at the Breast Solution website that Breast Cancer Trials has set up, which explores this in more detail. So thank you, and stop to take questions there.
Thank you, Geoff. And that's it reminds me of what we were talking about earlier with Caroline about the value of being able to take existing drugs. I mean, I wouldn't have predicted that something that's used to treat osteoporosis would have a benefit in potentially in breast cancer treatment. But I guess there's a library of medications that we can look at. Is that where some of your research is leading? Not just that drug, but other ones?
Some of it. I mean, you've heard about organoids today from Caroline, and we certainly have established organoid technology and other technologies where we try to barcode cells, understand their behavior at a single cell level through a variety of techniques, and where we can use libraries to identify targets. We've actually recently looked at BRCA2, another high-risk gene that can predispose to breast cancer, and identified everolimus, which is another drug that's already in the clinic to treat a number of conditions, including breast cancer, that could potentially be repurposed, or at least other types of drugs in that family could be used. It's interesting that you mentioned bone and breast. I guess I sort of naively think about the breast as being there to produce milk, and its role when it's a mature organ is to suck calcium out of the body and to generate calcium-containing milk. Of course, the bones are there as a repository of calcium for the body. So in a way, it's not surprising that nature hijacks common pathways between different tissues and organs that can very cleverly be used in ways that we would have never imagined until we do the research.
Yes, that makes sense. And I guess, looking at it from those different angles does give you different insights. One thing that this is probably a completely, unexpected and left field question, but one thing that I find really quite interesting about, your area of research is you're looking at stem cells in the breast and how they, develop through a person's life. But when you think about it, the reproductive organs and the breasts, in women particularly, they are among the most regenerative tissues in the entire body. Caroline mentioned it before with the endometrium, the lining is that it's very regenerative. Breasts go through changes in puberty, then through fertile life, they can go through cycles of dormancy, then to preparing during pregnancy and changing, and lactation, and then weaning, and then changes again through menopause. Do you think that those periods of rapid change and dormancy and that almost elasticity, is that something that is related to the tendency of breast to form breast cancer? Yeah.
Absolutely. I mean, it's something we've been really interested in for many years now. That the breast is one of the few organs in the body that develops postnatally. Most of the action happens during puberty and then pregnancy, and there is a massive requirement for ductal cells to develop into milk-producing cells during pregnancy. And so that has to be very highly regulated by stem cell as their daughter cells, and signaling between those cells and the immune system. And it has to be very carefully managed. For example, when women stop breastfeeding, because there's this massive disappearance of milk-producing cells. And so if that's not properly organised by the body and controlled, that has the potential for these cells to undergo genetic mishaps, hang around for too long and give rise to cancer. And we know, interestingly, during pregnancy, you can see very aggressive breast cancers occasionally arising, probably due to this highly proliferative process driven by the progenitor cells in the breast. Even for up to 10 years following a pregnancy, the risk of breast cancer is higher than for a non-pregnant woman or a woman who hasn't gone through pregnancy. But there is something fundamentally special about stem cell biology, so that in the longer term, the tissue changes and pregnancy actually protects against breast cancer, but there is this increased risk. And so understanding how stem cells and their daughter cells behave is really important to understand those cyclical processes that you mentioned, that happen through hormone signaling.
Thank you, Geoff. I'd now like to invite all of the speakers back to the screen. One of the things that I find great about the panel that we have here tonight is that we have people who are working on stem cells to understand what's the difference between normal development of cells and what happens when they go wrong, i.e., leading to breast cancer, for example. We've got stem cells being used to diagnose conditions, to understand endometriosis, and we've got stem cells being used to test drugs. So as a testing, like an alternative to a precursor to animal and human trials. And we've also got stem cells producing repair tissues. So that's a whole suite of different applications. We have a mountain of questions coming through, so I'll whip through them and I'll ask for relatively quick answers. Now, Sam asked earlier, for those people with prolapse and for purposes of treatment, is this going to allow for the regeneration of pelvic floor tissue to perfection, or what's a reasonable expectation?
So I think the main criteria is what we use clinically, the POPQ score, which is the POPQ quantification score. That is the clinically used measurements to say how bad the prolapse is. So I think what we would want is the reversal of the injury and a POPQ score where it is not prolapse. That is what we are aiming for. And I think the regeneration, once we have implanted, and in terms of birth injury, I think it would be reversal of birth injury, changes in signature of the tissue that is associated with injury, and then we give a treatment and it's reversed. So we have identified these markers or points of measurement that we would want to go back to. But the critical thing is to avoid complications and not go in the adversity path. We want to avoid that at all costs.
I think there's going to be a lot of questions for you, Shayanti. Yes, Ulla has asked, I might paraphrase some of these questions. So is the solution using stem cells only for treating prolapse, or would it also treat levator ani avulsion? So this might be Caroline, yourself, or Shayanti. Would either of you like to comment on that?
I think Caroline, you can
I'll have a try. Yeah. I'll have a try. As you can tell, prolapse is actually caused by a number of different things, or the pelvic support structures are the pelvic floor muscles, as Shayanti talked about, but there's also the vaginal walls, the tissue of that, and then there's the suspensory ligaments. And when we started this project, we couldn't look at all of the things. So we looked first at the vaginal wall, and that's what Shayanti's looking at now with the 3D printing and things like that. So we still need to address things like avulsion, because that's a really difficult area. And I know some of the urogynecologists have tried to actually stitch back the avulsed muscle, which is a muscle that's torn off, which they do for footballers, and not a pelvic floor muscle, but one of their quads or something. But it's been very difficult to do and hasn't really been successful. So I think that's going to need another big, massive input research and understanding of pelvic floor muscles. As Shayanti said, they are a little bit different to the skeletal muscles like your quads and things like that, that they do adapt to pregnancy, but they still undergo massive stretching and damage. And I can see from a lot of the questions here, this is the most difficult thing to deal with prolapse, is the avulsion, because it leads to severe prolapse straight up fairly early, and it's very difficult to do something about it. So we need a lot more research.
Yes, I'll just quickly add to that. This is a starting point. I would say we're just at the starting point, and there are researchers around the world who are looking at developing stem cell-based therapy and injectable-based therapy for avulsion. And one of the critical points or limitation that we have is we don't have suitable models. We can't access this tissue in form of human tissue. We do not have, enough animal models that replicate this. It's kind of like we're trying to treat something that we can't touch, see. So it's kind of very difficult.
I have noticed, though, that there are, just the other day, there was an article on, are we throwing away menstrual fluid that could be really useful source of cells in New York Times? And I immediately thought of your work, Caroline. I get the sense that we're having, hopefully, a bit of a renaissance with researching women's reproductive health in these tissues. Is that the sense that you're getting? Is there more interest from government funding, for example?
I think definitely in the endometriosis area, and I can tell you now, it's because the women with endometriosis have really become very active over quite a long period of time, and that's led to more funding being available. And I think it needs to happen for prolapse as well. And while I think endometriosis is a hidden condition, it's sort of coming out of the closet, prolapse is taboo. You can't really talk about it too much. And I think we rely now, and I'm pleased to see so many people interested and want to actually ask questions. We rely on you to actually raise the profile of this, because we will help too, because I think if the women who've got the problem speak up, and it's such a massive problem that, eventually funding will come. Because what we're doing has been so novel, and that we've had quite a lot of funding for the prolapse work, but it could do with a lot more and a lot more researchers working on it, actually. And so I just think any of you who are suffering from those conditions, avulsion or prolapse, where you can get active and talk to your parliamentarians and people like that, I really do think we can make a change and get more of a funding into this area.
True, very true. Well, you know what to do, audience. Geoff, what are the possible prevention measures for breast cancer in those that are at risk, the risk factors that you mentioned? Sam has asked that question.
Yeah, no, look, I think that's really important. And I focused on BRCA1 because that was our initial interest, to understand those at the highest genetic risk, if you like. But there's a lot of work that's been done by others, and work that we're interested in doing, looking at prevention. There are actually lifestyle factors that could be very helpful. We know that about 30% of breast cancers are potentially sort of preventable through lifestyle intervention. We know that exercise is very good, maintaining a good body mass index, all of those simple things. Unfortunately, alcohol is not a good risk factor for breast cancer, and the lower intake of alcohol is now strongly recommended, because that can be linked. Hormones are, of course, as we've discussed already, in one way or another linked to breast cancer risk. So avoiding unnecessary hormone replacement therapy, I think, is important. There is a drug called tamoxifen and raloxifene, which have been studied in the clinic many years ago now and proven to prevent hormone receptor positive breast cancer. There's with tamoxifen, there's something like a 30% reduction in the incidence of hormone receptor positive breast cancer for women who take it for up to 5 years. And that risk reduction carries over. The challenge with tamoxifen is that it hasn't actually been proven to reduce breast cancer mortality, but nevertheless, not getting breast cancer in the first place is often quite a good place to be for women. And so that's something that there are online programs that people can actually look at to determine their own risk. And there's one program called I Prevent, developed by Kelly Phillips, that people can actually ascertain their own risk and then go and see their GPs if they seem to fall into a higher risk category to explore some of these options. But for all cancers, we can probably all, do very sensible things like healthy lifestyles, avoid smoking, alcohol, and, maintain some level of fitness.
So there's no silver bullets. Is that what you're telling us? It's those
no,
yes.
But,
Yes, true.
There are,
Yes. Thank you, Geoff. And that's all we have time for. I think we could have asked questions for a lot longer. But thank you for your questions. But please join me in thanking our panellists, Caroline Gargett, Shayanti Mukherjee, and Geoff Lindeman. Now, if you or someone you know has questions about stem cell treatments that are currently available, we do have an information resource to help, because things like the treatments that Shayanti talked about are a few years away. There are stem cell treatments for things like blood cancers, but not everything that's advertised as a stem cell treatment on the internet is safe or effective. The good news is that the Foundation has produced a guidebook, What You Need to Know About Stem Cell Therapies, and that can be downloaded from our website at www.stemcellfoundation.net. And Kieran, my colleague, is going to post a link to the Stem Cell Therapies Handbook in the chat in a moment. While you're there at the Stem Cell Foundation website, I thought I might remind you that it's nearly the end of financial year. What a coincidence. So if you'd like to make a tax-deductible donation to the National Stem Cell Foundation of Australia, that would be lovely. And again, I'll get Kieran to put a link to the donate page into the chat box there. The Foundation uses donated funds to provide patient and public education and also to support promising Australian researchers. The scientists we've funded are working on everything from basic fundamental research to improving treatments for cancer, multiple sclerosis, cystic fibrosis, heart disease, diabetes, vision loss, osteoarthritis, and many more conditions. And you can read more about the scientists we've supported on our website. If you would like to find out more information about making a donation, then you can contact our General Manager, Graham Megan, via email, which is inquiries@stemcellfoundation.net.au. Thank you again for joining us tonight. Thank you again, Geoff, Shayanti, and Caroline. And we'll see you next time we have a webinar. Have a good evening.